Chapter 1, part 2. We're talking about themes. Themes connect the concepts of biology, and biology consists of many details. And if we can organize them into themes, then it is easier to remember. Earlier, we talked about in my previous video about properties of organisms at each level, and what is community, population, ecosystem, biosphere, and all that. We stopped at this point. So next theme is how in an ecosystem nutrients and energy work together to make life possible. So remember, in an ecosystem, there are abiotic components. That means they are not alive, such as water, air, soil, and biotic. That means living organisms. Um, in an ecosystem, living organisms utilize nutrients to survive, to function, and these nutrients they absorb from soil. After the nutrients become part of an organism, it will help the organism survive, function, and when they die, they get mixed into, or their body parts get mixed into soil, and the nutrients are given back to soil. So they are cycled. Nutrient cycle between organisms and its environment. Coming to energy, energy only flows. Energy coming from sun is going to support the organisms to carry out their functions, and that energy, after we use it, gets converted to another form, and it is just released from an organism and goes to environment, never comes back, or never goes back to sun either. So energy flows, whereas nutrients cycle. So this blue line, blue arrow, is showing how nutrients cycle, and energy only flows in one direction, from sun to environment, through organisms. So those organisms which produce energy rich molecules by incorporating or trapping sun's energy into their nutrients, they are called producers. Producers are photosynthetic bacteria and plants, whereas consumers are those organisms which depend on these producers to survive, such as animals. So animals are herbivores and carnivores. They both indirectly or indirectly depend on producers, which are plants or photosynthetic bacteria. So all the notes are here in the slide. Next concept, form fits function. What is form fits function? Form means shape, and function is how the living organisms move around or just carry out their life activities. So example would be a horse limb must be cylindrical, whereas bird's wing must be thin and flat. Can you imagine a horse having a thin and flat limb like a bird? No, it does not work, right? Because horses have to run, so they have to have a cylindrical, strong limb, unlike a bird. Whereas bird cannot have a solid limb like a horse. It does not work very well for a bird. Their limbs have to be nice and thin and flat for it to push the wind away, and also lighter. So that is form fits function. In biological organisms, the shapes of their body parts whether they're external or internal inside the living organism, which is a cell. Inside the cell, the organelles, at every level, the structure is modified in such a way that it will help their function. Next theme, cells are an organism's basic units of structure and function. This is the definition of a cell. Try to recollect the definition of an atom. Atom is the fundamental unit of matter, whereas cell is the fundamental unit of structure and function in an organism. There is a difference. So whenever you're answering question to do with fundamental unit, make sure that you're looking at the question properly and the choices, and read the question and choices completely. That's my suggestion. Um, a very short definition for cell. Cell is the fundamental unit of life. What is a cell? A cell is a structure found in living organisms, and it is enclosed by a membrane and has genetic material made of DNA. Every cell, whether it is bacterial cell or human cell, plant cell, does not matter what cell or which organism that cell belongs to, but it definitely should contain membrane, and it should contain genetic material, DNA. These two things are the requirements for any structure to be considered as a cell. There are two types of cells. Simple cells are called prokaryotic cells. Complex cells are called eukaryotic cells. You means you and I, right? We have eukaryotic cells. What are eukaryotic cells? Eukaryotic cells means they definitely have nucleus and membrane-bound organelles, whereas prokaryotic cells, karyon means nucleus. They do not have nucleus, they do not have organelles in them, membrane-bound organelles in them. So simple cells are prokaryotic cells, complex cells are eukaryotic cells. You and I are complex, and we have eukaryotic cells. Prokaryotic cells, example are bacteria and archaea. This is archaea spelling. They're both prokaryotic. That means they do not have nucleus or membrane-bound organelles. Whereas eukaryotic cells are found in plants, animals, fungi, and other small life forms. We'll see the pictures in next slides. Couple more differences between eukaryotic cells and prokaryotic cells. Eukaryotic cells are comparatively bigger in size to prokaryotic cells, and you can see here that eukaryotic cell has nucleus, which is nuclear membrane surrounding the genetic material, whereas prokaryotic cells have genetic material, but they do not have the membrane around their genetic material. Eukaryotic cells have organelles, prokaryotic cells do not have organelles. Do prokaryotic cells have ribosomes? Yes, they do. But remember, they are not membrane-bound organelles. They are just simple structures inside the nucleus. So you should know all the differences between prokaryotes and eukaryotes. Make sure that you know all these. The three differences that are described here, which is prokaryotes reproduce by binary fission, whereas eukaryotes develop and grow by mitosis and form gametes by meiosis. Transcription overlaps translation in prokaryotes. Transcription is separated from translation, and there is only one origin of replication in prokaryotes, whereas eukaryotic chromosomes have multiple origin of replication. These three differences we will talk about in later chapters in detail, and these are the things which you need in your final exam, for your final exam. So um, this is a picture showing us the development and growth of a human. This is a sperm, which is one cell, and egg, which is ovum, and they both contain chromosomes. Because this is human, there are 23 chromosomes in ovum and 23 in sperm. When they both fuse, they form a first cell called zygote. Zygote is made of two half cells called sperm and ovum, and this zygote is the first cell that is formed, and we all started our life with zygote. We were all one cell once upon a time when we originally formed. This one cell undergoes cell divisions called mitosis. It will undergo mitosis and it will form two cells. Those two cells undergo mitosis again, which is cell division, and they form four cells. Each one will form two and two. Like that, it will keep on dividing and will form an embryo. And this embryo, some of the cells decide to become head, and in those cells some of them decide to become left part of the brain, and others become right part of the brain, and some cells become eyes, so on and so forth. All the information to make a human baby out of this ball of cells is in the DNA. So remember, the DNA is the genetic material which is telling the cell when to divide, how to divide, and how many times to divide. And after they divide these cells, they get the information that they have to form so and so parts of the organism and finally make an infant. So this a part of life where we are we are becoming an infant from one cell, that is zygote, that is called development, and from fetus to adult, that stage is growth. So zygote to infant is development, infant to adult would be growth. DNA is the material which is in our nucleus, which is making chromosomes, controls the development and maintenance of an organism by a process called gene expression. Our genes, which are telling the cells what to do, when to do, they are expressed. So let's see, what is gene expression. Gene expression is a process of converting the information in the genes into proteins or protein products. So you have genes for making your skin color, hair texture, nose shape, your height, everything. They all come from the genes which were passed on to us by our parents. So what is gene expression? Gene expression is a process which is carried out by our cells, and in this process cells use the information in genes to synthesize proteins, and this process has two stages, or is made of two stages. Gene expression, the gene expression process contains two stages. First one is called transcription, second one is called translation. What is transcription? Information in our genes, that is DNA, is transcribed or copied, and we make a copy of a gene called RNA. After this stage, the information which is in the copy of the gene, that is RNA, should be translated into protein. So this is called translation. And all the genes are expressed through this two-step process called gene expression. First step is transcription, And second step is translation. um, I want you to remember one thing very clearly, that mm, each chromosome is one DNA molecule, and on one chromosome we have hundreds to thousands of genes. So yes, chromosome is made of DNA. So I apologize for this. Chromosome is made of DNA, and genes which are part of chromosome are definitely made of DNA. When chromosome is made of DNA, genes should also be made of DNA. Genes encode information for synthesizing proteins through a process called gene expression. DNA is passed on to the new generation um, by their parents, and DNA controls the development and the maintenance of an organism through gene expression. Every DNA molecule is made of two polynucleotide strands, and these two polynucleotide strands together make a double helix. Each polynucleotide strand in a DNA molecule is made of many nucleotides, and each nucleotide has um, three parts, and the part which is different in every nucleotide is called nitrogenous base, and there are four nitrogenous bases: A, G, C, and T. So look at this. This is DNA. Each chromosome is one DNA molecule. One DNA molecule is made of two polynucleotide strands. So looking at one polynucleotide strand, you can see that it has three parts: a phosphate, which is yellow color part, a sugar, and nitrogenous base. As you can see, that sugar and phosphate are all same in every nucleotide, whereas nitrogenous bases are of four types: A, G, T, C. So one chromosome is one DNA molecule. One DNA molecule has two polynucleotide strands. Each polynucleotide has three parts: phosphate, sugar, nitrogenous base. Nitrogenous bases are of four types: A, C, T, G. So these are the 1, 2, 3, 4s of DNA. One DNA molecule, two polynucleotide strands. A nucleotide has three parts. They are phosphate, deoxyribose sugar, and nitrogenous base. Nitrogenous bases are of four types: A, C, T, G. Try to remember this 1, 2, 3, 4 of DNA. Next thing, feedback mechanisms. What is feedback? When we are reflecting on what has happened and how it has helped us, and that is called feedback, right? So feedback mechanisms are found in biochemical reactions taking place in cells. What are biochemical reactions? Chemical reactions which are taking place in living organisms are called biological, or biochemical reactions. Biochemical reactions, when they are taking place, they cannot take place forever and ever and ever. They should be regulated. So how does a cell tell itself that do not carry out this reaction anymore? Let's take an example of a cell which is burning food, your muscle cell. Let's say, so your muscle cell is going to burn food more when you're exercising in a gym, but very less when you're sitting in a classroom. But then how does a cell know that you're not supposed to burn off the food when you're sitting in a classroom, whereas when you go from classroom immediately to gym, at that time it should start burning food? So this is by feedback. When a process or a biochemical reaction is taking place, and the product that is formed out of that chemical reaction is stopping that biochemical reaction, then it is called negative feedback mechanism. Whereas a product that is formed is actually causing that reaction to take place more, then this is called positive feedback mechanism. I want you to learn, understand, and take more examples for each of this process, because there is a complex question on this concept in the test. So look at this negative feedback mechanism. Let's take the example of formation of energy molecule called ATP. What is ATP? ATP is energy molecule which cells regenerate every time they need to carry out any physical activity. We are carrying out physical activity. Though we are not physically moving from one location to another, we are carrying out physical activity all the time. For example, though you're sitting still in the classroom, your heart is still pumping and your food is being digested and your brain is thinking, your kidneys are filtering blood. So many activities are going on. So you're burning food and making a lot of ATP, and ATPs are helping your heart to pump, and every function requires ATP. So this energy molecule ATP, which is formed by burning food, when they accumulate, so think that this D is a product of burning food. Food is A, and it is gradually burned but with the help of enzymes, and finally ATPs are formed. And ATPs, a lot of them accumulate because you're sitting still in a classroom and you're not using them up. Then these ATPs, they go and bind to this enzyme 1, which is actually burning food molecule A, and will tell enzyme 1 that, you know what, do not carry out any more chemical reaction, any more burning of food, because this person is not using ATP as of now. So that is how a process is going to tell itself, or the product of a chemical reaction is going to tell the cell, do not carry out this chemical reaction anymore. Such kind of regulation of a chemical reaction is called negative feedback mechanism. Example is synthesis of ATP, regulation of synthesis of ATP. Opposite is positive feedback mechanism. Example is a clot formation. What is positive feedback and how to understand? So whenever you have a cut, you start to bleed and your cell gets a signal that it has to make a blood clot. So blood clot is the product. And when a small blood clot is formed, the cell knows that it has to continue this process and increase the blood clot in order to completely seal that cut or wound. So formation of little blood clot will lead to more blood clot, and this is called positive feedback mechanism. So remember, product is stopping the chemical reaction from occurring by inhibiting the enzyme which is working early in the pathway, then it is called negative feedback mechanism. Whereas the product is binding to the enzymes and stimulating them to carry out more of the chemical reaction, then it is called positive feedback mechanism. Next concept is about evolution and organizing the different organisms for our understanding. There are 1.8 million species identified so far, and this number is going on increasing. There will be more species added as time goes. And all these organisms that have been discovered so far and are going to be discovered, they should be classified and placed in different groups. And this kind of branch of biology, or this kind of study which classifies the organisms into different branches, is called taxonomy. And there are different levels in a taxonomy, and to remember those levels, this is a very good mnemonic. Deer, let's not make it dove, let's make it deer. So, dear King Philip came over for good soup. So, dear King Philip came over for good soup. If you can remember this, then you can remember the levels in a taxonomy. So, our first one is domain. Under domain, there is kingdom. Kingdom is made of many phyla. Each phylum is made of many classes. One class is made of many orders. Orders are made of family. Family contains many genus, and each genus has many species. You have to remember this order. So to remember this order, the mnemonic is Dear King Philip came over for good soup. There could be many questions in this on this order, so remembering the order, the sequence is important for the test. You may answer this question and pause and answer the question. So each domain, which is the highest umbrella, just like in a company, there on the topmost there is CEO, like that. In biology, the topmost level in classification is domain, and there are two big umbrellas or two big domains. I'm sorry, three big domains. Domain Eubacteria, domain Archaea. They both are prokaryotes. Domain Eukarya. So these are the three domains. Domains are three: bacteria, archaea, eukarya. Eukarya is divided into four kingdoms, not three kingdoms, four. And they are Animalia, Plantae, Fungi, Protists. They are divided into four kingdoms based on their nutrition, how they eat, or how they get their energy source from. Can you see this one? This is amoeba. Don't think that amoeba is bacteria. Amoeba is not bacteria. Amoeba is eukarya. It contains nucleus. Bacteria are prokaryotes. They do not have nucleus. Archaea are also prokaryotes. They do not have nucleus. Whereas plants, animals, fungi, and protists, they contain nucleus and they contain organelles. Remember the difference. So you have to change this in your notes. Domain eukarya is divided into four kingdoms based on their nutrition, mode of nutrition. Next concept is evolution. Evolution is change. Evolution is the concept which explains how the different species might have originated. For some people, evolution is true. Other people, they do not want to support evolution. Either way, we have to study some concepts on evolution, and you're going to study further more about evolution, and you can have more discussions about evolution in biology too. Some things that are important regarding this concept is that DNA is a genetic material which is found in all living organisms. All organisms, either living or dead, but living organisms, that is what makes the difference between you and I and people and plants. And bacteria and eukaryotes, we all have different DNA, but we all have DNA. DNA is common, but in DNA, the kind of DNA we have is different among person to person, different among people and plants. Darwin's theory is also called theory of evolution. Fossils and some other evidence supports evolution. This is a very good example of how natural selection causes the change in the existing species. Read about this example in textbook. And unity in diversity, it um, it explains how we all have same internal structure, but externally it might vary. For example, bat, which is a bird-like animal, and humans, horse, whales, they're all mammals. Internally they have same skeletal structure, very similar DNA, but outwards we are different depending on the function. So we are getting modified depending on the function that we have to perform. We are not getting modified, but evolution occurs in population. Last concept is scientific method. In science, hard work alone is not enough. Sometimes luck, strategies, and being anti-fragile. We're not giving up. We have to keep going. Though we are facing failures, we have to keep on continuing the experiments, and have the hope that one day you'll be able to find the solution to the question, or your curiosity will be answered. Your question will be answered. That's how it should be. Remember what Vince Lombardi said: winning is not everything, winning is the only thing. So have that kind of belief, and attitude towards science in this class. Also, whenever going gets tough, remember that you have to keep going, and one day you'll be successful. In test, there will be questions on two main types of inquiry. What is inquiry? So learn all these concepts. Discovery science, two types of data: quantitative, qualitative. This is easy to follow from the notes. I'll leave this up to you and do not take more time. The video. 6 steps in scientific method are observation, question, hypothesis, experiment, analysis of results, and conclusion. Scientists are people who are curious. So when they observe things, they have question, and as soon as we have questions, we come up with immediate guess solutions or answers, and they are hypotheses. And scientists take their hypothesis one at a time and design experiment and get results and analyze their results, and results will help them either accept or reject hypothesis. Remember, there is no true or false kind of words in science. We are people who do not want to use such statements, because what is true on planet Earth might not be true on planet Mars. What is true for humans might not be true for plants or mice. So we do not use words true or false, we use words accept or reject. And we did an experiment, not experiment, activity in lab 1 on scientific method. So make sure that you know very well scientific method questions. There will be some surveys on scientific method that will be emailed to you. Be ready to answer those questions, and know the differences between theory and hypothesis. Theory is broader in scope, not hypothesis. Theory is general, and it may lead to more hypotheses. Theory is always supported by evidence, experimental evidence, scientific research evidence, and it will help us understand more and more in science, whereas hypothesis is just a statement or a guess answer to your question which does not have evidence. So these are some of the review questions for you. This is the end of chapter 1.